So guys, complement system. Why does it look so complicated? Okay, not to worry. I'll make it less complicated. Yeah, that's better. So, once again, complement system. So, if you are someone who do not know anything about complement system, well done. You are a normal human being. If you want to be abnormal, let's study complement system. Okay, so what do we have here? Certain basic questions if you know nothing about this complement system. First question is, what is complement system? Why is it called complement system? What is interesting about this system? And what do we have to know about this system at this level? At this level in the sense, if you are in Mbbs or something, okay? Or if you're just interested to know, not some in high depth research complicated stuff, just normal basic stuff. What is complement system? So what is complement system? They say it complements the immune system. What does it mean? It means, so our immune cells, okay, they produce certain proteins called as antibodies. These antibodies themselves goes and attack or attach on the pathogens, okay, and then these complement system do something which enhances their activity, and at the end the pathogen is destroyed or killed or eliminated or neutralized. So why is it called complement system? I just said it complements the immune system. It helps in destroying, killing, neutralizing, what else, whatever it is. So it helps the immune system to clear the pathogens. What is interesting about this system? The interesting thing is that the first thing is it kills the pathogen or the bacteria, or it neutralizes. Again, I'm repeating the same thing. Then it also helps in the inflammatory response. It also helps in opsonization. What is opsonization? It is nothing but if a bacteria has to be killed by the macrophages, especially by the macrophages or neutrophils, it has to be opsonized. So again, what is opsonization? It is nothing but, it is just like jam on the bread. So if there is bread, you are hungry, and if there is bread, although you can just directly eat the bread, but isn't it better for you to eat it with the jam on it? That means it just enhances the taste of the bread, and it will make you eat the bread so hard. That's true. So opsonization is nothing but the sugar coating or jam coating of the bacteria, so that either neutrophils or macrophages, they come and quickly eat them with full of enthusiasm. Okay, so like 20 proteins present which comes under the complement system ka proteins. They are around 20. Where are all this present? They are present in the blood vessels. That means in the blood. Where are they coming from? Are they coming from heaven, hell, or from trees? No, they are coming from the liver. Where? Liver. Okay, so liver is constantly making them and releasing them in the bloodstream. So like, what is the connection between these antibodies and complement system? That we will know in this lecture. So here is my B cell. B cell is transforming into plasma cell. Plasma cell is producing antibodies. Why is it gone mad to produce antibodies? No, because there is some infection, there is some pathogen, hence he is producing antibodies against that pathogen. So here is our bacteria, so beautiful bacteria, and here is our antibody. So do you think this antibody kills the bacteria? What do you say? No, no means no. This antibody doesn't do anything apart from building a machine which will kill the bacteria. So I also, until I studied the complement system, I thought these antibodies, they kill the bacteria or the pathogens, but the reality is different. This antibody, they do not kill the bacteria. It is the complement system that kills the bacteria. How does that happen? So complement system are like the parts, okay, certain protein parts which will come together to form something, and that will kill the bacteria. So what is the role of antibody? And the role of antibody is to tag or to get attached on the pathogen, that is in this case bacteria, and with the help of those complement system, it makes something altogether and then it kills the bacteria with something. So we will know what is that something and something. Okay, next. So this is just the introduction. I will suggest you to pause and give it a read, and then we'll move forward. If I read and explain you everything, it will take a long time. Video will be unnecessarily prolonged, so I'll just move. Okay, basic things. Complement system is a collection of plasma protein that functions mainly in the host defense against microbes. Yeah, obviously. And in pathologic inflammatory reaction, so it consists of more than 20 proteins and everything. So if you want to know what are all those 20 proteins, I'll just show you. So all these around 20 or more than 20, whatever it is. Okay, so this is it. Today we will be talking about this thing, classical pathway proteins. Okay, it is also called as classical complement pathway, right? So we'll go back. So classical pathway proteins, what does it mean? Why is it called classical? Because this was the first, when the researchers they discovered that okay, there is something which is killing the bacteria and it is not antibody, it is that that complement proteins. So that time they were like, okay, these are the classic proteins. Out of the 20 complement proteins, from C1 to C9, these are the, actually not C1 to C9, C1 to C4, these are the classic complement proteins, and this C5, C6, C7, C8, C9, these are called as MAC complex. That is nothing but membrane attack complex, also called as terminal pathway proteins. So we will memorize and understand this thing. Okay, so you just need to know counting to understand this. So C1, 2, 3, 4, 5, 6, 7, 8, 9. Out of this, you just have to know okay, 5, 6, 7, 8, 9. These are called as membrane attack complex. So that means something. I'll show you forward in next slide. Just read what is there. C1 complex composed of subunits C1Q, So C1 is called as complex. Why it is called complex? Because it is not just any one unit, but it has subunits. What are the subunits? QRS. So you just need to know 1, 2, 3, 4, 5, 6, 7, 8, 9. That is counting, and A, B, C, D, F, G, H, I, J, K, L, M, N, O, QRS. Okay. Then we have C2. Here is an exception. So before knowing the exception, come here. As you might have noticed, I have marked this as red, this one as red, and this one as red. Why did I mark? Because this has two components. Within them there are two components. One is the A component, the other one is the B component. So all these complement proteins, they are generally present as an inactive component in the bloodstream. Okay, they are inactive, they are not active. They are inactive because of that obvious reason कि we do not need complement system to get activated unnecessarily if there is no requirement of that. Until or unless it is not required, until or unless there is no invasion of the pathogen or anything, we do not need active complement system. Why? Because it will cause unnecessarily inflammation and damage our own tissue. So in a healthy individual, it is present as inactive, and that inactive is this whole thing, the A and the B. So this is the A component, this is the B component. Now what happens? So the red ones are those complement proteins which are having two components, like A component and B component, A component, B component, and A component and B. Okay, where is my marker? There's a pointer. So this has AB, AB, and AB. So what is exception in the C2? Come here. So as you can see, this is inactive. Now this inactive form, now if there is infection or anything, bacteria is invaded, so it has to undergo some process to become active. So C3, B, and A, this will become active. How? So the A component will go away. Okay, and as you can see, this is different size and this is different, this is big and this is small. So okay, again we just need to know which one is big, which one is small. So you just need to know what 1, 2, 3, 4, 5, 7, 9, 10. You just need to know ABCD, and you should be able to differentiate which is big and small. So this is just like nursery things. Okay, so which one is big? C3B is big. Which one is small? A1 is small. So A1 goes away and this big one comes this side and this becomes active. Now it is active. In books you will see there is a bar. In books when this active components will be written, there will be a bar on this. Okay, I forgot to mention it, but anyways you will understand. So this inactive has become active. How? The C3A component gone away B component is here. B is nothing but the binding component. Okay, this will bind and this will go away. This also has its own role. This has its own role. But I wanted to say you okay, C2 is an exception because in this the A component is larger. Generally in C3, C4, C5 the B component will be larger and this will bind to the complex of the whole thing. Okay, I will show you the what all the complex is forming there. But in C2 the A component is bigger and that will bind. Okay, so this was all in this slide to be known and to be understood. So again, you just need to be like graduated from the nursery class to understand what did I say till here. Next, classical pathway, antibody mediated IgG and IgM. So we know IgG is an antibody, IgM is an antibody, and so essentially it means this classical pathway is antibody mediated. That means we need an antibody prior, okay, to in order to activate this complement pathway. Which complement pathway? Classical, the first one which was discovered. Now see, I have arranged these in a certain way and I have put a drill machine here. What is this? This is a drill machine. Okay, so this is called as MAC. What was MAC? This is MAC, membrane attack complex, or the terminal pathway proteins, or C56789. 56789. So this is membrane attack complex. It's nothing but the drill machine. You have to remember that C56789 is drill machine. Then we have here this C1. C1 looks something like this in electron microscope or whatever, however they have taken this image, 3D image. This is a hexameric compound. Okay, 1 2 3 4 5 6. Why do I count? Because I don't trust them. So C1 is hexameric. Okay, and why? So after C1 we have C4, C2, C3, C5, and I have written something here called as C3 convertase, C5 convertase. Why did I write all this? Why did I make this markings? See, C1. So now what happens is, as I have told you already, out of all this, some has two components. Okay, like C2 was an exception. C3 has two components, A and B. C4 has two components. C5 has two components. C6 to C9 does not have that kind of component. C1 does not have. It is already a complex, QRS complex. Okay. Now let's bring a bacteria. Okay, and after this bacteria is entered into the body, do you think all this complement will go and attack on this? No. Why? Because I've already said that this is the classical pathway. What do we need in classical pathway? Antibody, because this is antibody mediated. So we need an antibody, and where is that antibody coming from? Here, from the plasma cell, B cell. Okay, so let's get an antibody. Okay, now we have the antibody. Now what? After attaching the antibody on this bacteria, now what will happen with all these proteins in the blood coming from liver? Okay, so see what happens. First, C1 is coming. Where it is coming? It is coming to this part is called the FC region. FC means constant region. This is the variable region where the bacteria is getting attached. Okay, and this one is the constant region. So in the tail of that antibody, or the constant region of the antibody, C1 is coming and attaching. Okay, actually what happens is, there are certain regions here. Okay, this will be hidden inside the antibody, and why this will be hidden? Because we do not need this C1 to come and attach here, right? Like in a normal healthy individual, until or unless this pathogen is not here, the region here will not be exposed outside. If it is once exposed outside, then C1 will come and attach. So after this bacteria is being attacked by this antibody, only then the certain regions here will expose itself outside, and once it is exposed, only then the complement C1 will come and attach here. And once this C1 is attached here, do you think this C1 has any components like A and B? No, it does not have. Which one has? C4 has, C2 has, C3 has, C5 has. C2 was an exception case. Okay, so C1 has come here anyways. Now it looks like this. Okay, then what happens? Then C4, actually I have shown here as C4 and C2, they are coming simultaneously, but it does not happen like that. First C4 comes, okay. C4 comes, and as I have showed you ki it has two component, A component and B component. B component is active, which binds, B means binding, and A component goes away. Similarly here also, when C1 comes, after that first C4 comes, okay. Do not get confused ki both of them are coming simultaneously. First C4 comes, it gets attached to the C1. How? It also has that A component and B component. B component gets attached and A component just goes away for a bit, okay. For a moment it goes away, then the B component binds to the C1. Then similarly C component comes, C2. Here this C2 A component, this was the exception, do you remember? A component was the bigger one and that one binds here. So this 3, when they bound here, this 3 is called as, overall this 3 called as C3 convertase, okay. C3 convertase means next what will come? C3 will come, okay. So C3 also had A component and B component. A component goes away and B component is binding here, okay. So all together this is called as C5 convertase. So what was it? Did you understand? This was antibody. First C1 was here, then C4, A component gone, B component got attached to the C1, then C2 comes, the A component attaches, B component goes away, all together called as C3 converters. Then C3 comes, the A component of the C3 goes away and the B component binds. Now all this together called as C5 converters. So obviously next what will come? The C5. C5 will come and attach here. Now after this C5 gets attached here, this whole thing again, like successively C6, C7, C8, C9, all this will come, okay. So C1 to C9 everything will be here, but this C1, C2, this thing was the initial part of the complement system, okay, the initial part, and this thing is called the terminal part, the terminal process. So I have made it all together as a drill machine. So after C3, C5 gets active, the C5 comes and attach here, and then C6, C7, C8, like successively they come. But in this animation I have shown as overall the MAC complex. That means the C5, C6, C7, this thing comes, this is a drill machine. Now what is happening here? So the antibody is attached here, and this has made a drill machine with this complement system. And what will this drill machine do? This will drill inside the bacteria's cell wall, okay. So will drill deep inside, and what will happen after drilling? Water will move inside. So not just one drill, this this drill machine, multiple complement system will come and drill everywhere. Everywhere it will drill, okay. So this will create multiple pores. After creating multiple pores, the water will move inside and there will be swelling of the bacteria and the bacteria will burst, okay. So did you understand anything? Did it make sense? C1, then C4, C2, C3. Actually you might have question ki why not C1 ke baad C2, then C3, then C4, then C5. Actually the thing is, the way these proteins were discovered was like this. First they discovered C1, then they discovered C4, then C2, okay. So there was a history between. They are misordered in numbering because of their discovering, okay. At different times they discovered different components of this complement system, hence it is like a little bit misordered. But anyways, you have to remember C1, then C4, C2, then C3, then C5, and after C5 C6, C7, C8, C8, C9 and everything, and this all together called as MAC complex, membrane attack. This is attacking complex. So it drills inside and then there is swelling and bursting of the bacteria. So if you see in any images and everything, you will see something like this, okay. And this was what? This was the classical pathway. Are there any other pathways? Yes, there are other pathways also, like alternate pathway. Then there is Manos pathway. So I think we will discuss that in some other videos, okay. Let's not make this single video too long. Okay, I'll just give a brief key why do we need alternate pathway. as I have told, in this classical pathway, it is antibody mediated. That means against this bacteria, your body needs to have a prior or already existing antibodies against this against this antigen or bacteria. So what will happen if the bacteria is new or any new brand new pathogen is entering in your body? Do you think this classical pathway will act on this? No, right? But we have this complement system. Our body does not want ki it will just sit quietly, it will do nothing if any new bacteria is entering. No, our body has other mechanisms in which they will use all this MAC complex in other ways. So that is an alternate pathway. Okay, that we'll learn in some other video. So these are all the 20 proteins of that complement system. You just pause and give it a read. And this thing also you might be understanding now. This is the classical pathway. Okay, see your pathogen antigen, then see QRS, C4 is coming, C2 is coming, all together is forming C5, actually C3 convertase, then there is C5 convertase, then C5, all this 6, 7, 8, 9, it is forming something called as MAC complex, which is the drilling machine. And these are the other pathways. Okay, this is the Manos pathway, this is the alternate pathway. We'll discuss this in other videos. This is given in Robbins classical pathway, antibody mediated. Okay, so yeah, then this is C3b, 3A component goes away, MAC complex. And these are the other functions of the complement system. One is inflammation, then there's phagocytosis, nothing but the opsonization, and the lysis by the MAC complex. Okay, complement system. Okay.